Ladies and gentlemen, I'm pleasure to invite Professor Krzysztof Liberek to say today's webinar. Professor Krzysztof Liberek graduated from physics at Technical University of Gdansk, received PhD in molecular biology and habilitation in biological sciences at the same university. Krzysztof Liberek carried out postdoctoral work at the University of Utah and at the University of Geneve in Switzerland, working with Professor Costa Georgopoulos. Krzysztof Liberek is a full professor since 2002. In 2006, he was selected to EMBO. He is interested in the role of and mechanism of chaperones proteins action in different cellular processes, and today he will talk about chaperone proteins. Krzysztof, the floor is yours.
So, proteins are not particularly stable molecules, and in stress conditions, they get partially unfolded and they aggregate, forming either amorphous aggregates or fibrillary structures. And here you can see that, for example, in bacterial cells, in cell poles, the aggregates are formed, and this can be also analyzed using fluorescent-prone proteins, which at stress conditions form fluorescent foci at cell poles. And it was shown that approximately 5% of the total protein inside the cell aggregate in stress conditions. So you can see it here, starting from 45, you can see substantial aggregation in bacterial cells. Similar phenomenon you can observe in yeast cells. So at 25, nothing is bad, but when you go to 44, you can see electron-dense structures which correspond to aggregated proteins. And also in human cells, it was reported by Chris Dobson laboratory that expression levels of human genes are remarkably anti-correlated with the aggregation rates of the corresponding proteins measured in vitro by the experiment. And this result indicates that the human proteins has evolved to resist aggregation and to function efficiently, but with almost no margin of safety to respond to genetic or environmental factors, which decrease their solubility. So, and we all, of course, know that neurodegenerative diseases are associated with protein aggregation. All organisms have developed the mechanism which allowed them to fight with the aggregation problems. In stress conditions, the synthesis of chaperones proteins is substantially increased, and the heat shock response during which the chaperone proteins are produced was discovered in 1962 by Italian researcher Federico Ritossa. And he observed that specific paths are formed on chromosomes of Drosophila melanogaster in heat stress conditions. And in this area, the efficient translation processes were taken care, were observed. However, this observation was forgotten for more than approximately 20 years. And in the 80s, it was reported that when you increase the temperature of the larvae of Drosophila, then the expression of synthesis of constitutively expressed proteins is shut down. And what is synthesized inside the cell, only heat shock proteins are synthesized in these conditions. And but not all heat shock proteins are molecular chaperones. And molecular chaperones are defined as a large and diverse group of unrelated proteins that all share the functional property of assisting the non-covalent assembly and disassembly of protein-containing structures in vivo, but are not permanent components of these structures when they are performing their normal functions. And chaperone proteins are divided into families: HSP100, HSP90, HSP60, called also chaperone, HSP70, and its co-chaperones HSP40. It is J-domain proteins and small heat shock proteins. And today I will talk about small heat shock proteins, HSP70, 40, and HSP100. So chaperone proteins bear several functions in cells, both in physiological conditions, housekeeping activities. Mostly they play important role in de novo protein folding, protein translocation across membranes, assembly and disassembly of complexes, and also they play a role in the proteolysis. But today I'll talk about the functions of chaperone proteins in stress conditions. And in stress conditions, chaperones are involved in prevention of protein aggregation. They are actually doing the disaggregation process, and they are also important for refolding of aggregated proteins. And today I will concentrate on these three functions, particularly on protein disaggregation. And in 1985, there was a paper by Hugh Pelham, in this paper in Cell, he suggested that during the heat shock response, proteins become partially denatured and they expose hydrophobic regions which then interact to form insoluble aggregates. By binding tightly to hydrophobic surfaces, HSP70 changes its conformation and limits interactions with and promotes disaggregation. So, and then 4 years later, there was a paper from Maciej Żylich laboratory, which is also published in Cell. And in this paper, they show that bacterial HSP70 is able to reactivate heat-inactivated RNA polymerase in ATP-dependent manner. And this is the original curve from Maciej's and Dorota's paper. And you can observe here that you need quite a lot of DNA K to refold one molecule of RNA polymerase, more than 100. But later, I was also involved in this research, were able to show that when you add co-chaperones of DNA K, means HSP70, J-domain protein, and nucleotide exchange factor, you can greatly decrease the amount of HSP70 which is needed to refold heat-inactivated RNA polymerase. And actually, we were able to show that it is the sigma 70 factor which aggregates during the heat stress, and the rest of the polymerase is functional. So only sigma 70 factor was reactivated in these conditions. More or less at the same time, there was work performed by Susan Lindquist on yeast cells. And she showed in 1990 that gene coding HSP104 chaperone is crucial for developing the thermotolerance. And the thermotolerance is defined as the ability of cells to survive the heat shock after previous exposition to stress conditions, so called pre-shock. So wild-type proteins, wild-type cells were dying, but not so efficiently. When you have delta HSP104 strain, the difference is quite big, since this is the logarithmic scale. And Susan Lindquist also showed that at stress conditions, the proteins inside these cells do aggregate. However, during the recovery, the aggregates disappear in wild-type cells, but they are present in delta HSP104 strains, suggesting that somehow HSP104 is involved in either disaggregation or proteolysis of proteins from aggregates. At the time, it was unknown. And in 1998, it was also a paper published by Susan Lindquist group, and they showed that HSP70 and HSP70 and HSP40 form a novel system that rescue previously aggregated proteins, and it was really very potent system capable of disaggregating aggregated proteins. And this is a system present in yeast cells, but analogous system is present in bacterial cells and also in plant cells. And HSP104 is the workhorse of the disaggregation problems. It is a hexameric protein, which the hexamer is not symmetrical. It's kind of spiral with the central pore inside. And the monomer of HSP104 is built up of two NBD nucleotide binding domains, 1 and 2. And it is 8AA plus protein. N-terminal domain, and characteristic for all disaggregases, long coiled domain. And this long coiled domain is outside of the hexameric structure, and it forms a ring on NBD1 domain. And what are the protein substrates of this disaggregase? It turned out that proteins aggregates cannot be disaggregated by HSP104 alone. HSP104 is able to translocate non-aggregated, intrinsically disordered proteins on its own, but it's not able to disaggregate aggregates on its own. So when we have GFP aggregates, HSP104 is not working. When we are adding HSP70 to the system, HSP70 system itself is also not functional. When we combine both systems, we are getting efficient refolding of luciferase, of GFP from aggregates. So what is HSP70 actually doing? The classical cycle of HSP is presented here. And first, HSP40 binds to misfolded substrates and delivers the substrate to HSP70 and triggers ATP hydrolysis by HSP70, and then HSP70 and ADP forms a complex with the substrate, a J-domain protein dissociates, and then nucleotide exchange factors is acting on this, and HSP70 goes to the ATP form. So knowing this, several laboratories suggested the roles of HSP70 system. And actually, work of several laboratories suggested that first, HSP70 binds to aggregates, and then it attracts these aggregates to aggregates through interactions, since HSP70 interacts with this coiled domain of HSP104, and it resulted in binding of HSP104 to aggregates. And HSP70 might be also involved in protein refolding downstream of these aggregates. So the polypeptides go through the central pore inside the hexamer, and then it has to fold downstream of this hexamer. And here you can see the monomer of HSP104, and this is this regulatory coiled domain. And when the coiled domain forms a ring on the NBD1 domain, the disaggregase is in the inactive form. And when HSP70 interacts with the disaggregase, the coiled region is moved away from NBD1 domain, and the protein is activated. And when we started our work on HSP104, by molecular dynamics studies, we observed that in this region, there might be a cell bridge before lysine and aspartic acids, which might be important for regulation of HSP104. So we introduce here lysine instead of aspartic acid, and now we have the repulsion, which should lead to the activation. And in fact, we observed this activation, and this protein was much more active than wild-type HSP104. However, to our surprise, this protein was also active in the absence of HSP70. So somehow this disaggregates this hyperactive mutant, which was in the repressed position, was independent of HSP7. So from the biochemical point of view, it's easy to create disaggregates which is more active. But when this disaggregate was put into these cells, it was toxic to the cells. So somehow interaction with HSP70 is important for the regulation. So it's not, so you have tightly control the disaggregates. So it's not only a problem of disaggregation, but the biggest problem is to control the disaggregates. And in our paper, we were able to show that another layer of the control of HSP104 is related to ADP level in these cells. So in normal conditions, the level of ADP is enough high to block all the activities, to inhibit all the activities of HSP104, namely substrate binding, ATPase activity of this protein, etc. And HSP70 promotes HSP104 binding to aggregates, but not to intrinsically disordered proteins. And when HSP104 binds a polypeptide, these aggregates, a translocation occurs and inhibition by ADP is overcome. And ATPase and all activities of HSP104 are restored. This is because when translocation occurs, the affinity for ATP is greatly increases, and ADP is no longer a potent inhibitor of this protein. And this is another layer of the regulation. So in normal cellular conditions, this protein, the activity of this protein is off, and without HSP70, it's basically dead. And recently, we become interested in how the system works. And for our studies, we use biolayer interferometry. So in this experimental setup, we have a protein layer on the surface of the sensor. And when the proteins bind to the sensor, there's a change in the interference pattern, and we can have real-time data on protein-protein interactions, analyzing the changes in the interference pattern of the line. So here is the idea of experiments which were performed by us. We have aggregates bound to the sensor, and then we are adding HSP70 system to it. So we can observe the binding and HSP104, and we can also observe the binding and then the dissociation step. And we recently become interested in the role of J-domain protein in binding, in Chaperone bindings to aggregates, since these proteins recognize the substrates and attracts HSP70 to aggregates. So here you can observe binding of HSP70, SSA, this is yeast SSA1 protein, and we have two yeast cytosolic J-domain proteins, CIS1 and YDJ1. And with CIS1, we observed this is the curve when CIS1 was present in interaction. So substantial delay in binding, and then substantial increase of the signal. And this is binding of the proteins when YDJ is present, so fast binding to around 2 nanometers. Binding of require ATP, where there is no ATP, there is no binding. So this is as expected. So in this point, we can observe there is more SSA associated with the sensor when as compared to YDJ. This is the quantification. So 2-fold more HSP70. So what are the consequences of this? More HSP70, and then we asked the question how HSP104 binds to this. And it's clearly when we have more HSP70, we are getting a higher signal for HSP104 than when we have YDJ on the sensor. And this is the comparison by Western blot and analysis of this. And this might be the reason why the reaction containing cis1 is more efficient in disaggregation as compared to the reaction containing YDJ in and HSP70 and HSP104. So we are also what was also very interesting for us. It was this delay in the binding, and when we were looking at this, we postulated that HSP70 system might have an additional role in disaggregation, namely aggregate modification. So we thought that the delay in binding might be due to the aggregation modification process which takes part and here. So actually we performed several experiments. I do not have time to talk about this, but actually we came into the conclusion that when more HSP70 molecules are bound to aggregates due to entropic pooling effect, the structure of the aggregates is changed, and then it's easier for the disaggregates to disaggregate and thread the polypeptides from the aggregates through its central channel. So this might be the other reason why this reaction when cis1 is present is more efficient compared to the reaction where YDJ is present. So J domain protein is important for the disaggregation process, and we published this last week in PNAS. There's also a very interesting problem related to the importance of the refolding of proteins from aggregates versus disaggregation of proteins from aggregates. Both processes formally result in the removal of aggregates from the cells. And the beautiful paper published by Bernd Buchau laboratory answered to this question. They were working on bacterial chaperones, so CipB is HSP104, bacterial HSP104. So what they did, they created a BAB version of CipB, and this BAB version of CipB was able to interact with CipP protease. So any polypeptide which was translocated through the central channel of this version of CipB was degraded. And then having this type of construct, then they perform in vivo experiment. So when they have delta CipB cells, there is no disaggregates present, so the aggregates are inside bacterial cells. When the wild type version of CipB is present, the disaggregation occurs. When BAB version of CipB is present, it's working like wild type CipB, so also disaggregation occurs. When BAB version of CipB was present and also additionally the protease was present, there are no aggregates in cell after 60 minutes. So basically, in all cases, the aggregates were removed from the cells. But then they ask how the cells survive this heat stress. And delta CipB and BAB CipB cells do not survive the stress in comparison to CipB and BAB containing cells. So this beautiful cell paper showed that actually it is important for the cell to disaggregate and refold active proteins from aggregate to survive. It's not enough to proteolyze aggregates because it is then the aggregates are not present anymore, but the cells are done. And up till now, I was talking about disaggregation in plant, bacteria, and fungi cells. And in this organism, there is disaggregase present. However, in humans, we do not have the disaggregase. We have HSP70, J-domains proteins, nucleotide exchange factors. So this is the difference. And a few years ago, it was reported that HSP70 system alone in metazoan is able to disaggregate aggregates on its own. So this system does not need disaggregase, which is present in yeast, bacteria, and plants in non-metazoan. So how it works? Well, it was suggested that first J-domain proteins binds to aggregates, then it recruits at HSP70, this is the classical cycle, and then HSP110, which is the nucleotide exchange factor for HSP70, binds to this complex. And then binding of such a big complex generate pulling forces due to entropic pulling. And the polypeptides, the single polypeptides are released, and they have the chance to fold. And last year, it was a paper in Nature showing that HSP70 system is able to disaggregate fibrils formed by alpha-synuclein. So the scheme of the reaction is basically the same. J-domain proteins recognize the fibrils. It has to be J-domain from B class. Then HSP70 is attract, and then HSP104 comes, and due to the entropic pooling results in disaggregation and folding of monomers to proper structure. And it was published last year in Nature. And up till now, I was talking about the disaggregation principles. So what we have aggregates, and then we disaggregate, proteins from aggregates, either with HSP70 system alone or with HSP70 system and disaggregates. However, there is another family of chaperones named small heat shock proteins, which are also important for disaggregation. However, they are not involved in the disaggregation per se, but they are involved in the aggregation. So they modify the aggregation process. And we were also interested in this process. So small heat shock proteins, of course, possess low molecular mass, 12 to 43 kilodaltons. They possess very conserved alpha crystalline domain. So they form large oligomers in non-stress conditions. They are widespread in all kingdoms of life. They are activated by stress conditions. So in stress conditions, they deoligomerize and they form dimers. And their chaperone activity, they stabilize protein aggregates in refolding competent cells. And for example, in E. coli, they are named EBPA and EBP2 from inclusion body associated proteins. So you can see here that when you have inclusion body, EBPA is associated, it binds to these inclusion bodies. So this is how they were discovered, and they form heterodimer. And here you can see the luciferase, which was heat denatured in the absence of small heat shock proteins. And here you can see the luciferase, which was heat denatured in the presence of small heat shock proteins. So the aggregates are completely different. And when we try to recover the luciferase from this or this type of aggregates, when we are adding HSP70 and HSP100 system, this is bacterial system. So when small heat shock proteins were absent, we are refolding approximately 15% of initial luciferase activity. And when small heat shock proteins were present during the denaturation step, we can recover 3 or 4-fold more luciferase activity from this type of aggregates comparing to this type of aggregates. And this small heat shock proteins has to be present during the denaturation step. When they are added at reactivation step, nothing really happens. And the luciferase is inactive in both types of aggregates. You can see it here. And we are also involved 2 years ago, actually, we published the paper in EMBO, and we show in this paper that HSP70 displaces small heat shock proteins from aggregates to initiate protein refolding. So after formation of the aggregates, small heat shock proteins form dynamic complexes with substrate, and they associate and dissociate from the substrates. And then HSP70 has to compete with small heat shock proteins for binding to aggregates. They has to win the competition, the protein, and then the disaggregation process can start. So in my talk, I was trying to present you the principles of disaggregation of the proteins which are formed in stress conditions. And here I presented our publication from last 6 years. And about some of them I was talking during my very briefly about during my talk, and some of them I was not really mentioning them, but this is what we published on this topic recently. And the people who were involved in the project, so Szymon Ziętkiewicz initiated work on disaggregates in my laboratory. Then Agnieszka Kosowska is working on HSP70 and HSP100 dependent disaggregation. Igor Obuchowski is working on small heat shock proteins, and Hubert Wyszkowski is working on J-domain and on the role of J-domain proteins in disaggregation. And Wiktoria Stangierska is involved in the project related to the nucleotide exchange factor in disaggregation. Piotr Karaś and Claudia are doing their PhD projects which are related to small heat shock proteins. And research in my laboratory was financed and is financed by the National Science Center, and previously were funded by the National Foundation for Polish Science. And thank you for your attention.
Thank you, Krzysztof. It was a great lecture, and we have answers and questions system you can use to ask the question. If you do have any, please click on the answers and questions or pytanie I odpowiedzi, and write down your questions. I will happily pass them to Krzysztof. And he will for sure answer. Okay, the first question is the system HSP170 involved in protein disaggregation when the aggregates results from other stress factors, example, oxidative stress?
Well, I think that this system is specifically designed for disaggregation of the aggregates formed by heat. And actually, it's tempting to speculate that these aggregates is present in the cells which are sessile, so they cannot escape from the heat, like plants, fungi. And so this is therefore disaggregate gene is important. In, for example, humans, this gene is missing, so disaggregation was probably not so important. And HSP70 system is doing its work. It's enough to do the work. So I think that rather it is not able to disaggregate. However, at least this is what I think about this. Or it's not involved in this in physiological in normal situation.
Thank you. Any other questions? Okay, there is another question. How many drugs are developed for disaggregation? Are there any efficient way to deal with disaggregation?
Well, actually, there was papers suggesting that HSP104 can be a weapon to combat diverse neurodegenerative disorders. So actually, there was maybe not the drugs, but there was the idea. There is still an idea that you can, by genetic therapy, you can introduce for example, or yeast or modified yeast disaggregates into, for example, cells. And yeast disaggregates is able to cooperate with human HSP70. And then efficient system for disaggregation can be created inside human cells. So this system is quite potent, and actually it was shown that it's working in tissue cultures or in yeast cells. But of course, no experiments were performed on living human beings.
Okay, there's another question. Does biolayer in interferometry you established your experiments also work for other protein binding domains like group 2 chaperones?
Yeah, yes, of course. Actually, we developed this. It was quite difficult to develop the system at which we have aggregated proteins on the sensor. Actually, this technique was developed to analyze interaction between the proteins. So you can have different type of sensors, either HISTAC, either you can have, for example, antibodies on the sensor, biotin, so several sensors. So actually, this technique was developed to analyze protein-protein interaction. And the aggregates is a specific case, and it was quite difficult to actually have the aggregates, to develop the method to bind aggregates to the sensor.
Okay, are there any other questions? I can't see, so thank you for the lecture.
Thank you.